Hello and welcome to another episode of Momento Malum today. The low bush blueberry is a blueberry species native to the eastern North America region. This species has a long utilitarian history. It is a landscape plant that's quite cold hardy, has delicious fruit, and has leaves that make good tea. It is also used extensively as an ornamental plant, mostly for its red fall foliage. This species is grown commercially in Maine, Quebec, and the Canadian Maritime provinces. The methods of farming low bush blueberry is unique among the fruit crops. The fields are not planted with cultivars with known traits or genetics. Instead, the forest is cleared and managed with fire and other methods to promote the spreading of existing populations of wild low bush blueberry, which are then cultivated. There is great interest in the gene pool of the low bush blueberry, and Dr. Lee Beers was one of the scientists who attempted to quantify the full genetic diversity of the species. And it's from this paper that we'll derive this video. The size of the low bush blueberry gene pool is relatively unknown compared to other fruit crops. In more recent years, it was discovered that the populations of low bush blueberry are highly diverse in both Maine and the Canadian Maritimes. The low bush blueberry is believed to be the first blueberry species to be managed in North America. Clearing and burning of forests by American Aboriginals for harvesting purposes occurred prior to the arrival of Europeans. Although attempts have been made to improve the farming systems or create commercial cultivars, the primary methodology of farming low bush blueberries has remained mostly unchanged for centuries. The farmers rely entirely on wild plant populations pre-existing in fields. The primary assumption that the researcher was testing was that plants in managed fields for commercial harvesting retain the same level of genetic diversity as plants growing in mature forests. This, however, may not be the case. In rabbit-eye blueberries, all cultivars are derived from 4 founder genetic lines, making a very shallow gene pool, a perennial problem for plant breeders. Existing low bush blueberry populations may have undergone a similar process. It is speculated that large events of mass destruction of a forest could result in plants descended from a smaller number of parents, which would create a genetic bottleneck. To determine the genetic diversity of cultivated low bush blueberry populations, 7 populations of both managed and unmanaged fields of blueberry were analyzed across multiple locations ranging from USDA Zone 4 to Zone 7. These populations would be analyzed genetically using the EST-PCR genetic technology that was developed specifically to analyze species in the genus Vaccinium, which includes blueberries, bilberries, cranberries, and so on and so forth. And here are the results. If you want to donate to my channel, the links are below. If you want to buy my book, you can also find that link in the description box below. The genetics showed that the low bush blueberries in managed and commercial fields do not retain the same level of genetic diversity found within old-growth forests. Although the fields of low bush blueberries do not contain clonally propagated cultivars, the management system removes some of the genotypes from the landscape. Fire kills a portion of the genotypes, and this is further compounded by the addition of the use of a common herbicide called hexazinone to wipe out weeds within the burned down forest that still linger on. Low bush blueberries also managed in a 2-year cycle, with a cropping year followed by a pruning year where the fields are mowed or burned. This will remove even more genotypes and populations, and this explains why there's a great deal of genetic loss within these managed fields. There is, however, still a great deal of genetic differentiation between cultivated populations, which is common across most Vaccinium species. The genetic diversity in the wild populations in comparison to the cultivated populations is far wider. and there's no evidence of inbreeding depression across any of the wild populations, and plausible evidence of crossbreeding in between diverse populations. And that about covers everything. Thank you for watching.